Hello, everyone, and welcome to today's live broadcast, Primary Cell Systems, Offerings and Applications from Basic Biology and Tissue Modeling to Compound Screening, presented by Dr. David Kuninger and Dr. Rhonda Newman. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's webinar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box on the left side of your screen and click the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click the Support tab found at the top right of the presentation window, or click on the Answer Question box and report your problem. We have two presenters for you today, Dr. David Thomas Kuninger and Dr. Rhonda Newman. For detailed biographies on our speakers, please click on the Speaker Biographies tab at the top of your screen. So without further ado, Dr. Kuninger, Dr. Newman, you may now begin your presentation.
Great. Thanks for the introduction, Christy, and we'll just get rolling. So, and again, thank you all for for dialing in or clicking in. It's really our pleasure to share with you our primary cell portfolio, some key applications, and yeah. So with that, I'm on the second slide. So yeah, this just outlines an overview of our talk. So first really is, what is the portfolio? What do we offer? And then a highlight of some key applications that we thought would be interesting in terms of the different ways to use primary cells to study various aspects of biology and different signaling pathways utilizing various technologies from the Thermo Fisher Scientific broad portfolio of technology and instrumentation, and then end up with Rhonda speaking specifically on using primary cells for creating organotypic skin models and some of the important applications those are used for. So, this slide 3 shows an overview of the cell types that we offer, and I should say these are, they span various tissue types, developmental stages. Principally, they're of human origin, although we do have a few non-human cell types. Principally, these are going to be provided as cryopreserved cells. Excuse me, although we have a few examples or a few offerings of proliferating cells and certainly flexibility to provide live cell cultures if that's needed on a custom basis. Slide 4. So, really the key to being able to develop and offer a broad portfolio of primary cells is access to tissue. High-quality tissue is really critical. This slide highlights, not meaning to gross you guys out, but where the cells are derived from. So this is typically comes from two major sources, which are indicated in the second bullet on the right. So we work closely with tissue procurement networks and organ donation groups, as well as local clinics or medical centers. And really there's two, for tissue that would be, potentially transplantable, the use in humans for transplant for life-saving or extending applications is always, primary use of those tissues and organs, but often, or at least with some regularity, one can't align donors, appropriate donors with those tissues. So to prevent them from going to waste and being able to leverage these very valuable tissues and gifts from individuals. We, along with other providers, are lined up to accept these within a reasonable window and then isolate the various primary cell types from them. On the other side of things, there are some tissues that are just discarded as part of normal medical practices. The upper right image shows a umbilical vein. So these are where we isolate HUVECs from. So it's typically not saved, discarded as part of the birthing process. The lower left image shows actually a fairly good-sized chunk of skin that was removed from an elective cosmetic surgery procedure. So again, various sources that we get these from. We have extensive networks on how to source, ages, different donor profiles. This is all a highly regulated space. We're compliant with federal and European guidelines for compliance with confidentiality, with HIPAA, with procurement and use standards. We can also provide some institutions or uses. It's important to have traceability back to donor consent. We can provide donor consent forms for each source tissue as needed. So I've mentioned thus far kind of different types of cells, sort of where we get them from a tissue standpoint, but really to get the most value out of the cells, it's really a cell system that's required. These again are specialized human cells, and as such, they typically require specialized or purpose-designed culture conditions, and this is something we spend a lot of time to develop and put together. So this slide really just highlights that concept, is that we have not only the cells that I showed, but basal media that's designed for those cells or cell types. Supplements that are purpose-built for the maintenance of sort of maintaining the physiological characteristics of the tissues from which the cells were derived, and other reagents for general culture, expansion, cryopreservation. So it's really, we look at it holistically and really try to provide these types of complete solutions. So slide 6, this just provides maybe a little bit more of a specific example of what a system could look like, and also I think highlights a couple other aspects. So often we have a range of options in terms of if you want, for example, a keratinocyte, a skin epithelial barrier cell, we can we have adult, neonatal, or pooled. Even within these, if there's some specific donor traits, we can, sort of select for those, or you could look for those. So there's sort of a range of cells. In this case, with the keratinocytes and the basal medium, we have a range of basal media and supplement combinations. And these can be, really from our most robust, and I guess robust would be, that would maximize the growth conditions. This typically means it has the highest protein content and likely contains undefined products, excuse me, such as a serum, or in the case of the keratinocyte supplements, the animal products here I'm referring to as bovine pituitary extract. So they're serum-free but have a pituitary glandular extract as part of it. However, if you would want to have more defined conditions, if that's important for particular assay endpoints or applications where you really want to have a more defined culture condition, again, we offer that with two different supplements. In this case, for example, our EDGS supplement or the S7, which are neither of which contain the bovine pituitary extract, but are either defined or completely animal origin free. And as a kind of a general rule of thumb, as you get to more defined culture conditions, generally have lower protein content, and also the need for extracellular matrix support typically goes hand in hand with using defined conditions, particularly for the primary cells. So in this case, I'm highlighting the use of a coding matrix. This is actually a recombinant human collagen type 1 solution that's really required for the effective culture in the defined conditions. So this is, again, how we've worked all of these, sort of options out in advance. I won't go into, sort of great detail with the other cell types and media options, but you can go to our, primary cells webpage and see that. Okay, so yeah, I touched on matrices for the keratinocytes. They typically, the dermal keratinocytes like collagen. That's the sort of their natural environment, or at least by and large, they attach well to collagen. We offer a range of other matrices that are shown here that are either, again, different flavors depending on your particular requirements. For 2D culture. And then we have two matrices listed below, the alga matrix and Geltrex. These can be used for both 2D or 3D culture. And I'll give you an example of some 3D culture on the next slide. And these are, I guess I'd say, maybe more classic applications of 3D culture. Using Geltrex and primary cells. So the upper set of images are an angiogenesis tube formation assay. So we actually sell a kit for this also if you're interested in it, but essentially these endothelial cells, in this case the HUVEC, human umbilical vein endothelial cells, or other large vessel endothelial cells would work well in this also, are seeded onto a Geltrex-coated plate. And they'll spontaneously form these tubule structures over time, and you can use this as a sort of classic model of angiogenesis. Has its, I think, both strengths and weaknesses, but clearly it's an application with the Geltrex. Another shown below. This is using an embedded culture. So if we look at the left two images. We're looking at human mammary epithelial cells cultured in 2D or in a 3D Geltrex gel. And so in this case, what we see are structures, these acinar or tubular-like. This model is designed to help mimic ductal-like structures that occur in the breast from which these cells are derived from. So this is an example of using, translating these cells from 2D to 3D with this Geltrex matrix to recapitulate structures that are seen in vivo with these cell types. And actually, you may notice these sort of spheroid structures that are, the slide on the lower right indicates these are fixed H&E cross-sectional slides where you can see they're actually sort of hollow balls or acinar structures. And these cells adapt a spheroid or myo, more of a fibril cell type too, depending on the cell ratio. So you can kind of see that in the middle lower slide. I would mention too that another key application for this material Geltrex is in the use of organoid culture. This is an area that's become has seen rapid growth over the past couple of years, and it's really gone hand in glove with the stem cell world, stem cell differentiation in 3D, and these matrices like Geltrex are really a typical and critical component to forming these large organoid differentiated structures. So just a little bit of a different application, but a key one for this matrix. Okay, so again, teeing up, I guess, stem cells a little bit. There are, so both Rhonda and I guess, we didn't, without reading our bios, we spend a lot of time in both the stem cell world, iPSC, culture and differentiation, as well as the primary cells. And, as such, I think we have a fairly good perspective on, or we've gained a perspective on the value of these cells in various applications. And so I thought it may be useful to put a table up to sort of capture some of at least my thoughts on the differences and where you may find value in one system versus the other. And I think there are clearly strengths and weaknesses with both systems. That is taking normal human primary cells or their sort of stem cell derived counterparts. And so I listed a few of the key ones here. And so I think right off of the bat, and this may be familiar for many of you folks on the call, but so during and particularly with iPSC, so induced pluripotent stem cells, there's an epigenetic reset during the process, and so and the cells that come out of the reprogramming and subsequent differentiation from following reprogramming tend to be fetal-like and early fetal-like, and so this can be a challenge if you want to study a number of a phenomenon that happens in adults, for example. So, whereas we can, one can isolate primary cells from individuals of different ages and look at age-related, characteristics or features. And indeed, we offer a number of cell types from a neonatal skin and adult skin from various ages. Looking at those types of, age-related studies in stem cells is difficult to impossible to do right now. Similarly, if you're looking at complex disease phenomenon, inflammatory diseases, mechanisms of disease that have a lot of have epigenetic or multi-genetic components that can arise over time or with sort of environmental influences, these can be very, very challenging to model with stem cells. And again, here's an area where primary cells are really probably ideally suited. Purity, it really varies. I mean, I'd say typically high in primary cells because we've had a lot of time to work through isolation conditions. With stem cells, it's variable. I think it is getting better. A lot of the differentiation protocols are getting more efficient, but so that's still a work, and that field is moving and progressing. So range of cell types, I think we are more limited with primary cell types in general. This can be, from sourcing, from isolation. There can be just real challenges with getting these certain cells and certain cell types. Stem cells, in principle, unlimited. You should, in principle, be able to derive, but in practice, it's not unlimited. It's not unlimited, and you can see with the expansion capability. Is, again, in principle, unlimited with stem cells. Primary cells have a Wilson S. They have a lifespan, and that's part of what makes them normal, but can also be a challenge. Okay, so moving on, again, just to speak a little bit to the workable window with primary cells, and just in terms of how many cells, what can I expect in terms of growth, how many experiments could I do with this, what's the relative, how much can I do with a single vial of primary cells. And so what we put this table together to try and address that somewhat and give you a sense of a scale. And so this table lists, on the left, 6 different primary cell types that we offer. I put the first column over is the average. So this is our typical production scale, average lot size. And then the next is sort of our minimum population doublings. So what's the minimum lifespan? And this is something that we QC our cell products for. So when you get a vial of cryopreserved cells from us, there's a certain viability, there's a certain cell number, and there's a guaranteed minimum expansion capability, and we typically will measure this in population doublings, not passages. They certainly correlate to each other, but population doublings a more accurate way to measure cell lifespan. So here we're just looking at the various cell types, and if you were to get a vial of these, expand them through their guaranteed lifespan, how many cells would you end up per vial? And you can see these are at minimum in the billions of cells, so it's really or more. And if you translate that to a certain number of plates, we used a 384 well plate format here, but you can see you can really, you're thoughtful in how you plan and expand and use these cells. There's really quite a bit of usable material even in a single vial. Okay, so I'm going to move into some of the application data. A good chunk of it is going to be leveraging our Bac Mam technology, which is highlighted on this slide. So Bac Mam is really it's co-opting a baculoviral, baculovirus, a recombinant baculovirus, to efficiently deliver genes to mammalian cells. We're on our second generation of the Bac Mam technology now, and the baculovirus, you see the cartoon image on the left, it's been engineered to have improved tropism and delivery to cells by tinkering with the coat proteins on the surface of the virus, and the expression cassette has been optimized for high-level sustained expression in mammalian cells. It's really easy to use. I'll go through the workflow. It's biosafety level 1. It's safe. We see effectively no toxicity in all practical ranges. It has a high cargo capacity, so we can get close to 38 kb or so. I think it's the largest content we've delivered. You can transduce cells with this and freeze them, make large banks of cells that you can pull out and use. I should mention that this is also transient. What we found in general is that the relative expression levels of your transgene of interest are similar to that for transient transfection. So again, it's a transient system. So workflow, it's really very, very simple to use. Simply just have your cells, sprinkle some virus on them, or add the virus. We have specific guidance on volumes or particles per cell, or relative, sort of titers or MOIs to use. But it's very simple to even do this from volumetrically. Typically incubate overnight, and you can start to image or quantify or use the cells the following day. Again, to give you a sense of kind of timing and dosing, this is looking at delivery of a baculovirus encoding GFP, an untargeted green fluorescent protein, to human epidermal keratinocytes at different doses, so this would be different volumes of the viral solution here, or particles per cell, but it's very simply done volumetrically. And then looking at the relative expression at one of these at a fixed volume. In this case, we're looking at, I think this was 400 particles per cell or so. And looking at expression of GFP over time. So if you look at the right set of images, the column with the green, so we're looking at GFP expression starting at 6 hours at the top and going down to 24 hours. The same field, phase contrast images are shown below. And then we're looking at, if you sort of go back to the graph, we're looking at, at this MOI, by 24 hours, we're seeing effectively, 80+ % transduction efficiency. So really efficient, really easy to use. We found this to be a very nice reagent for content delivery across a wide variety of cell types. So I'm going to touch on those right now. So here we're looking at Bac Mam. Again, we're looking at, this is a different cargo, and I'll talk about some of that in the next couple of slides. Oh, for some reason, I do not see the mouse cardiomyocytes up here. I don't know if that's showing up on your screen or not. But essentially, I'd say we've really looked across the board at a number of human and rodent primary cell types, and we get very efficient and very easy delivery, similar workflow, highly efficient delivery across a range of cell types. So the next slide is going to talk about the different types of content. So this is an effective delivery vector. So we've built out a range of different types of targets or assays, sensors that one can use here, and also you have the ability to customize. We provide custom services for building out viruses. You can also, we have DIY kits now for these viruses too. So it's, there's really a range of options for generating, either getting off-the-shelf reagents, having us make them, or making them yourself. The first set of, this slide is going to look at, so I'm going to talk about, just give you examples of all of these areas of content, and I think it's to highlight the types of experiments one can do within primary cells using these reagents. So the first is our cell lytes. A portfolio of products, and these are very simply RFP, they're RFP or GFP fusion proteins that have targeting sequences fused to them that will deliver and retain the fluorescent protein in the indicated compartment. So it's a very simple and effective way to image organelles or cellular structures, in a dynamic way. So this can be all, you can fix the cells and do other stuff, but you can also do live cell imaging. If you're interested in trafficking or dynamics that may, with whatever treatment or biological context you're looking at it, if there are dynamic changes in any of these structures, this is a very nice way to track, quantify, and image over time. I won't, again, this is just a slide showing different flavors of those targeted GFP or RFP constructs. Again, they work really well. They're very easy to use. We have a really nice, there's some online tools on how to pick, what do you want to look at? What other floors do you want, if you wanted to do multiplex imaging, what's the right sort of set of reagents to look at? Another one of the Bac Mam encoded types of reagents that we've developed and offered are those that are more specific to interrogating the activity of distinct signaling pathways, signal transduction pathways within the cells. So the first slide I'm just going to kind of tee up. This is another one of our cell products. This is a cryopreserved human skeletal myoblast. The cartoon shows it above, that you can plate these cells, and within 48 hours, they rapidly differentiate and form multinucleated myofibers. So, giving you a very rapid and robust human skeletal muscle differentiation system. What we're showing here is that we can deliver the baculovirus, we can apply the Bac Mam reagent to these cells and very efficiently deliver the GFP to the myofibers. So that's indicated in the sort of the 15, so the upper right ICC image. So this is GFP expression in the myofibers. The images below show same field. We're looking at nuclei in blue, or troponin. So we're looking at a marker of these mature skeletal myofibers in red. So very, very efficient delivery. So what these Bac Mam pathway sensors enable you to do is to look at distinct signaling pathways in the appropriate biological context. And so in this case, we're showing, so in muscle cells, skeletal muscle in particular, the IGF-1, insulin-like growth factor 1, and TGF-beta pathways are really critical for differentiation, for muscle maintenance, for maintaining a healthy muscle during disease conditions, muscle wasting, cachexia. So they're important pathways in muscle. And so what we're showing here, these are on the lower left. We've added one of our pathway sensors for the PI3 kinase AKT pathway. This is a lanthoscreen PRAS40. So essentially, this is a phosphorylation target that we're adding to the cells encoded by the Bac Mam. And what we're reading over time is the phosphorylation at threonine 246 in this PRAS40 following IGF stimulation. And so this is our assay is going from very little IGF to more. And these are physiologically relevant levels of IGF-1. This is sort of nanogram, sub-nanogram per ml levels. Similarly, when we look at the TGF-beta, either TGF-beta-1 or related family member myostatin using a phospho-SMAD3 lanthoscreen assay, we have, again, very nice sensitivity at physiological levels for both of these growth factors. So again, we can study the in this case, the pathways of interest with good sensitivity in the right type, in a relevant biological background. Similarly, I'm going to switch to a different Bac Mam sensor, and this is one that we can use for intracellular calcium sensing. So this is the Aequorin encoded Bac Mam. It's delivered to cells very much like the same workflow as I've shown before. The Aequorin, in this case, we have to load up with colentrazine. That's the CTZ, little pink molecule in the cartoon about halfway through. Once the cells are primed with this, you can provide an agonist and look for calcium signaling following agonist or antagonist, typically extracellular receptor binding events or activation events. This is very sensitive. It's sort of a flash-based fluorescence that has a very high signal-to-noise ratio. I mean, there are robust chemical sensors one can use also, but this system does work very well for high sensitivity and very low signal-to-noise. This is just showing some data with that system. In this case, we're looking at delivering the Bac Mam Aequorin to two different primary cell types. On the left, this is the human aortic smooth muscle cells. On the right, it's the human umbilical vein endothelial cells. And then we're looking at these are dose responses release of calcium and activation and the fluorescence or luminescence of the Aequorin following histamine stimulation. And so in this case, we're utilizing the endogenous receptors on the cell and putting basically a positive control ligand and stimulating the cells. And you can, if you look at the y-axis, this has a very broad or tall assay window. Similarly, we set up. We were curious about, how would one compare a similar type of screen. So we're looking at chemical library screens on either a workhorse cell line. So this is the top panel. This is a 293 cell that's been transduced with the Bac Mam Aequorin, or we're looking at a primary cell. The human aortic smooth muscle cells that that had the Aequorin delivered, and then we're looking at the top panel. The top graphs are the 293s. The upper left is one library plate of agonists. The other upper right is a separate antagonist plate. These are commercially available libraries that we were using. Below, it's the same layout, but we're using the human aortic smooth muscle cells. One thing we realized right away is we had good assay windows for both. We have pretty similar Z primes. We also had clear hits that are positive and negative controls both were as expected on this. Although what was interesting here is that we actually, if we look at the different compounds, the discrete hits were different in the workhorse line versus the primary cell line. And so, again, the context in which you are asking your biological questions matters, and so this just gives flexibility to do, again, compound screening in primary cells. Okay, so I'm going to just quickly move on through a couple other slides, and this is really just addressing some of the other. We within Thermo Fisher Scientific have a broad array of cell health reagents. These can be used in a wide array of biological contexts. They're fairly simple endpoint measurements. I've listed, again, here the modes of altered health, so we can measure simply. These are almost all image-based tools for measuring apoptosis, autophagy, necrosis, cell proliferation. We can drill down and look at mitochondrial health, mitochondrial membrane potential, ROS species. There's a whole range of them. What we found broadly is they translate very nicely into the primary cell applications. I'll just touch on two of those. So one, this is using our ClickIt EDU assay. This is a surrogate for, it's actually a replacement for the BRDU measurement for cell proliferation. It's very easy to use. We found it's highly compatible with the primary cell types we've looked at. It has just a much easier and simpler workflow than BRDU, more sensitive. Similarly, here is this is a Bac Mam encoded reagent it's our FUCCI Cell Cycle Sensor, and really what this encodes two different a red fluorescent protein, a green fluorescent protein that are fused to either the protein domains that are sensitive to cell cycle dependent ubiquitination and turnover. So essentially, you deliver both of them. The red protein is stable because of the CTD fusion domain is stable during G1, but rapidly becomes ubiquitinated and turned over in SG2 and M. So it really is its expression. It's the protein, not expression. The stability of the protein is dependent on the cell state of the cell cycle. Similarly, the Gemini EGFP fusion, the Gemini protein is rapidly ubiquitinated and degraded during G1. So you essentially don't have green GFP expression during the G1 phase of the cell cycle, but it's maintained throughout SG2 and M. So again, if you need to do live cell imaging of if you're looking at synchrony, or if you have events that you may think are cell cycle dependent, it's a very sort of easy and simple way to visualize the state of the cells. Okay, so with that, I hope I gave you a sense of our overview and some of the, I think, interesting tools and reagents for primary cells. I'm going to hand it over to Rhonda to take a focus dive into the organic using the developing organotypic skin models.
Yeah. Thanks, David. So the workflow for today's talk, basically I'm going to be talking to you about how we can implement the cells and different Thermo Fisher media reagents together to generate 3D epidermal skin models. And I'll be showing you how some of our plastics technologies with the tissue culture plates and their carrier plates really facilitate this stratification effort with minimum hands-on time. And briefly, I'll touch on how you could develop more sophisticated models via co-culture. So the first question is, why would you want to have 3D skin models? And obviously, when we go to think about different things in development of consumer products or even for understanding basic biology, there are a number of facets. So one of them being predictive toxicology. So it's well known that 3D epidermal skin models and full-thickness skin models can also be important for assessing the irritancy and corrosivity of various compounds. It can also be used to get a better understanding of transdermal drug delivery. And furthermore, it can be really important in product development, so particularly in the cosmetic space, where people are looking at different skin creams and lightening and darkening reagents, and obviously in other areas of facilitating wound healing. So today I'm going to take you through a do-it-yourself protocol that we generated using the Thermo Fisher reagents and describe that a bit to you. So first you'll start either with a proliferating culture, or you can use a cryopreserved bank of 2 to 3' human epidermal keratinocytes. And in this instance, you can use either neonatal or adult keratinocytes. And then you would feed those into polycarbonate membrane inserts and add medium to the upper and lower chambers. Then this submerged culture growth continues for a total of 2 days. And then by changing the media condition such that you raise the calcium chloride concentration and also you raise it to the air-liquid interface such that it is no longer fed within the insert, but rather just from the bottom up, this is known as raising to the air-liquid interface, and this continues for 8 to 10 plus days. And at that point, you get very nice stratification of your 3D models. And we have carried this out all the way up to 23 days and see very nice maintenance of these structures up to that point. So overall, the protocol takes 2 to 3 weeks, and it's a very versatile protocol. Some of the things that I called out, I'll highlight here. The protocol is available online as well at the URL link shown there. So as I mentioned, it is very flexible in that you can use either neonatal or adult. You can also start from a proliferating culture or cryopreserved bank, and here you can see we've started from, like, this particular example is from a 3' hex, so you have quite a bit of expandability so that you can get a large number of models. And I'll show you later how you can couple this with the NUNC inserts and carrier plates to reduce your hands-on time. And as I mentioned, it's important to ensure that you're varying your media condition between the submerged culture and raising to the air-liquid interface, as the increase of calcium chloride is important in the stratification process. So I want to take you through a few of the properties of these do-it-yourself 3D epidermal skin models. First, we're going to look at the histology. So the image on the left, we're highlighting a full-thickness skin explant. And what you can see is you'll see the epidermis there on the top, and the dermis is underneath, and it's comprised of fibroblasts rather than keratinocytes and a large collagen composition. So as we're only focusing on the epidermis for this do-it-yourself kit, you can see here's a representative 3D skin model from that, and I'll just slowly point through the layers. So here we have our polycarbonate membrane that it is cultured on, and there are a wide variety of different membranes that cells can be cultured on, and different cell types like different membranes. And again, in this instance, polycarbonate is the choice. You can see the progenitor layer of stratum basal, followed by stratum spinosum and stratum granulosum. And then finally, you get your outside surface of the skin known as the stratum corneum. And that will grow as the cells become more aged. So overall, we see that we have a very appropriately stratified epidermis generated using this protocol. Furthermore, if we look at immunocytochemistry, looking at markers of differentiation in these skin models, we see very nice correlation with what we see with the skin explant. So here, looking at epidermis and dermis, a reminder that in the explants we have both compartments. So for keratin 10, this is meant to highlight the progenitor layer, the basal layer, and you can see in our epidermal model, it is right along the surface of the polycarbonate insert and mimics very nicely what you see with the skin explant. With keratin 14, this is an early marker of differentiation, so it's not in the it isn't in the basal layer of cells in the stratum basal, but rather in the layers above, and you can see its nice expression in correlation with what we see in the skin explant. Invitrogen is an intermediate marker, and so you can see it spread a little throughout, and it correlates very nicely with what we see with the skin explant, whereas flagrin is a late marker of differentiation. You see that in the outer surface of the cells. So overall, our 3D epidermal skin models also have very nice marker expression. So as I mentioned earlier, predictive toxicity is a key area for use of these 3D skin model equivalents. So a couple of different things that are important there is the barrier properties, and these can be assessed either using an ET50 measurement, which I'll go through with you now, as well as transepithelial electrical resistance, or TER measurements, and I'll briefly mention those. So in an ET50 measurement, what you'll do is you'll take your skin model and dose it topically and incubate it for a range of incubation times, and I'll step through some of that data. Then you copiously wash the model, and then you'll transfer it to a dish containing a viability reagent, and there's a number of them available. The MTT is a validated viability reagent for this given purpose. You can also, we've also tested things like XCT, Almar Blue, Presta Blue as well. But in this instance, using MTT, as you incubate it for 3 hours in that solution, basically the live cells will turn purple due to the generation of the formation crystals, whereas the dead tissues will remain opaque. And finally, you'll extract that color from them and measure the absorbance at 540 nanometers. And here's just some representative data showing what to expect. So in this instance, you will have water as a negative control, and that should be added at the longest treatment time in your testing scheme. So here, since we went out to 9 hours, our negative control is tested at 9 hours. We also would have a positive control. Here we use 5% SDS, and you can see that it has a large impact on the viability of the cells. And then subsequently, if we look at 1% Triton X100, you can see that there is a time response to this, where at 7.5 hours, it's at roughly 50% viable. So these 3D epidermal models do demonstrate good barrier properties with an ET50 of around 7 hours. And furthermore, if you were to look at the transepithelial electrical resistance, depending upon the model that you derived, it could be anywhere between 4,000 and 7,000 ohms per centimeter squared. So now I want to talk to you a little bit about coupling of our cells and media with the NUNC cell culture inserts and carrier plates, and this is a very nice development that reduces the amount of hands-on time. Basically, you take these NUNC polycarbonate inserts and you can put those into carrier plates, and they're available in both 12-well and 24-well formats, and they lock into these carrier plates. And one of the really nice things is that it can lock in at various positions. So you can have it at a high, medium, or low position, depending on the amount of medium and the frequency of feed that you want to attain. So this is a very nice complement, and it does allow for less frequent feed schedules. Another thing I did want to mention is that the NUNC inserts are also available in 3 different pore sizes, and this allows you to have flexibility in your research, because not only are the NUNC cell culture inserts important for things such as the epidermal skin stratification, but if you are interested in cell invasion assays or other types of things, then those are available to you. So here we just want to show, using the NUNC carrier system, how it affords flexibility in the feed schedule. So again, there's the 3 different insert positions. As you raise the insert position, obviously you need to include more medium in the lower compartment. And what this affords is that you don't have to change the medium as frequently as in the protocol. The original protocol that we worked off of, we were doing feed strategies every other day. Here you can skip up to 4 days. So if we look at the overall stratification of these models, here's at the low position, you see nice stratum basal, stratum spinosum, stratum granulosum, stratum corneum, and the same for the middle position as well as the high position. So it does show the appropriate stratification for all three positions. So now I just want to lightly touch on how you might achieve a more sophisticated model by implementation of the human epidermal melanocytes, and we do offer these in a range of pigmented types from moderate to dark pigmented human epidermal melanocytes. And so in this particular instance, we have a control, which is 100% human epidermal keratinocytes from adult, or a pigmented in which we've added 7 per 93 HSKM. And so you can already see the darkening in the bottom, but as you add L-DOPA, this will cause that to magnify, the darkening to magnify. And if you look at the overall stratification, you can begin to make out some of the melanocytes that are contributing to the darkening of the layers. So this is one way in which you can, a cold culture method in which you can increase the flexibility of the system. So as I mentioned, the protocol is available online, and here's just a snapshot of, we provide the cells, media, and supplements, with the exception of 2 of the reagents. They're all available. And we've also tested this with the presence of the antibiotic and antimycotic, which again is optional, but it's nice to have that security blanket for long differentiation periods or stratification periods. And then also I highlighted the use of the polycarbonate inserts in the carrier tray. So with that, I think David took you through David and I both took you through a diverse range of primary cell offerings that we provide at Thermo Fisher, and also discussed a bit about how we can couple that with the different media and matrices that we offer. We can offer off-the-shelf solutions, but also custom of even animal origin or defined culture medium and reagents. So definitely reach out to us if you have questions on that front. And then we also highlighted specific examples where you compare together our media and cells together with a plasticware to further expand to physiologically relevant endpoints. So we want to thank you for your attention in today's presentation and open up the floor to questions.
Thank you, Dr. Rhonda Newman and Dr. David Kuninger, for your great presentation. As Dr. Newman mentioned, it is time for our live Q&A portion of the webinar. If you do have a question you'd like to ask, please do so now and click on that Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Are you able to obtain other cell types? I don't see my cell on your list.
So possibly, and I think, so this is an area we have, so first we have a specific form that we can, you could go through your Thermo Fisher sales rep for, or find it online. What we found is it can often be cost prohibitive to go, I mean, just to be completely transparent here, it may depend on how much you want. Is really, because if we don't currently make it, we're going to have to spend some time on the isolation condition. It can be a little bit time consuming and costly for us to go after other cell types, very different cell types that aren't listed. So if it's something that you really need, and you need it to run a program, or you would anticipate, let's say more than 10 vials, or a substantial volume of cells, it would probably be worth discussing. If not. It may just be cost prohibitive for us to do that. I would say that what we often do is we'll go after a different form. Yeah. So maybe I'll just leave it at that.
Okay. Well, Dr. Kuninger, I'm going to piggyback on that question, and I have a more specific one asking, do you provide osteoblast, osteoclast cells?
No, we don't. We don't currently provide them. I mean we have MSCs. Oh, so sorry, as Rhonda was mentioning, or sorry, sitting here. No, so formally we do offer the progenitor cell type from which they are derived. So mesenchymal stem cells we have in a couple of flavors. So that's probably as close as we could do. So we don't have the osteoblast or osteoclast per se, but the progenitors from which they're derived.
Okay, thank you. Next question, two-part. What methods have you found to be the most successful in transfecting primary cells? And do other non-viral methods work?
Yeah, that's a really good question. So I'd say as a general rule of thumb, electroporation is a very effective method to deliver nucleic acid content to primary cells. So we offer the neon system, although I know that wasn't the question specifically. It's a little bit of a caveated answer. So we have transfection. I'm going to assume we're talking about delivering mid-sized DNA plasmids here, but so we have a range of transfection reagents that have that I'd say work well to okay in primary cells, depending on the cell type. So that would be our lipofectamine 3000, or even the earlier generation lipofectamine 2000. But what we do provide is on the website, and I don't know the URL off the top of my head, but you could look up lipofectamine and transfection guide, and there's a very extensive table that's listed on there that would provide, the guidance for the reagent and DNA ratio, so some overall transfection, what you might expect to see, what we would recommend, and some general transfection guidance there.
Yeah, and to piggyback on what David said, the neon electroporation does work very well, and there's a very large table that covers a very diverse range of cell types that can be effectively delivered. In the neon, the nice thing about it is despite whether you're delivering protein, nucleic acid, it delivers pretty much everything. So much more flexibility there.
But that requires someone has the instrument, instead of just a consumable that the transfection reagent is.
Okay, thank you. Here's another question. How about capillaries in 3D skin models?
So we've not worked on that per se. As far as we've gone is just with the introduction of melanocytes. Obviously, as you get more sophisticated models, you could start putting in dermal equivalent. I've not seen great success with capillaries, being in those systems at this time.
Okay. Now, what is the greatest challenge in developing a 3D model of human skin using this system?
Yeah, so one of the really great things about the human epidermal keratinocytes, and one of the things that was highlighted in one of David's slides, is the expandability of them is really great. And so it's not so much that cell source. The one thing that I'd say is keeping an eye on your inserts, that you're not getting any air bubbles trapped beneath them. So that can sometimes be tricky, because obviously you want the media to be able to go all the way through the bottom of the insert. So I would say that that's a tricky part. Also, if you're wanting to make more complicated models, such as full thickness skin models, while you can embed fibroblasts into rat tail collagen, if you're looking at wound healing applications, not all fibroblasts are equivalent, so there may need to be additional screening factors that are considered there.
Okay. How about, here's one that just came in. I'm focused on CRISPR-based knockouts. Have you done anything with CRISPR and your primary cells?
We haven't done, well, I don't know about the custom biology team.
No, we have, this is a very interesting question though. We're looking at, we have a number of tools, yeah, so CRISPR-based screening tools that are encoded by lentiviruses. And so we think that would be, we would expect to have good delivery with those vectors into these cells. As Rhonda mentioned also, the neon electroporator, yeah, we know we can effectively deliver, well, we have more data with stem cells, but right, we know we can effectively deliver a variety of, protein, RNA, the protein guide complexes, or plasmid-based CRISPR and plasmid-based Cas9 and CRISPR guides effectively also by electroporation. So yeah, I think it would. We don't have the data here, but yeah, it should lend itself to that either way.
Yeah, and there a lot of times when working with pluripotent stem cells, you use like a HEC293 as a positive control to say that your tools are working. In that instance, I realize it's not a primary cell, but it should be very similar. And there's a new offering, a TrueCut Cas9 nuclease, which is really great because it has a more efficient nuclear localization targeting signal on it. There's also synthetic guide RNAs that you can order that are custom and deliver away. So, but again, the neon, I think, is the best option there.
Thank you, Dr. Newman, and thank you, Dr. Kuninger. We're almost out of time, but would either of you like to leave our audience with any final comments?
I mean, I'd just say, again, for those of you who are still with us, thank you. And thanks for attending. I hope it was useful. We did have contact information for Rhonda and myself or our business partners, and we're happy to have any, we're here doing this all the time. We, happy to handle any follow-up questions or discuss anything that comes out of this. So thank you.
Thank you.
Thank you again, and thank you for your important research. Before we go, I also want to thank our audience for joining us today and for their interesting questions. Any question that we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. I also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through September 2018. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thanks again for joining us, and we hope to see you again soon. Bye-bye. And we're clear.